Hi there, I'm Harveen. I am an fourth year undergrad student currently pursuing Bachelor of Science in Biotechnology degree. It's a joint degree at Holland College and University of Prince Edward Island. Recently, I did a research study. I was investigating the effects of curcumin on the regulation of TP53 and BAX gene expression in lung cancer cell line. So the lung cancer cell line that I used throughout this research, it was a 549 cell line. So basically, talking about a bit of the background, um, curcumin is extensively used in Asian countries. It's an active ingredient. It has been used for like over thousands of years. Previous studies have shown that curcumin shows apoptotic effects, even inflammatory effects in cancer cell lines. So I have chosen two genes here, TP53 and BAX. So the reason why I chose them, it was pretty much related to their apoptotic um, effects that they have. So TP53 it encodes for another protein which is called P53, that's also known as like the tumor protein. So TP53 gene, it basically takes care of repairing DNA. It tries to repair DNA if it finds any mutation, but if it cannot, then it basically causes apoptosis, like the self-destruction of the cell, to prevent any kind of infection. So I chose TP53 gene due to this effect. And the other gene I chose was the BAX gene. It is a pro-apoptotic member of the BCL2 family, and it basically encodes for BAX alpha protein. It has shown that BAX, when it is linked to an activated BCL2 protein, it basically causes apoptotic effects in cancer cell lines. So both of the genes that I'm researching on, they both should upregulate upon the treatment of curcumin, which is a turmeric extract. This is what I'm hypothesizing. Um, and there should be basically a decrease in cell viability, like the cell survival, um, should decrease upon the treatment of curcumin with the upregulation of these two genes is what I'm expecting. So the methods that I basically used in this experiment, they involved extraction of curcumin from turmeric using Soxhlet extraction. And then it was quantified by UPLC. Um, I did UPLC analysis to check the amount of curcumin that I'll have in my extract and calculation of recovery rate. I basically cultured my A549 cell line, which is the lung cancer cell line that I'm using, and treated it with different doses of curcumin. The treatment was for two different timelines. One was 24-hour and the other one was 48 hours of the curcumin treatment over the lung cancer cell line. The third thing that I focused on, it was the gene expression of TP53 and BAX genes upon the treatment of curcumin in the lung cancer cell lines. So talking about my results, um, so basically when I ran my extract on UPLC, I got six different peaks. Five different peaks are for unidentified compounds, but one peak is for curcumin, which matched the curcumin standard. So it had like highest value in terms of area percentage, showing like a good amount of curcumin was present in it. So the curcumin extraction was overall successful, but with a low recovery rate. Like I got a recovery rate of 1.7%. But still, we can say that the curcumin was extracted. Now, the second thing, moving forward to cytotoxicity of curcumin, like I was testing it using MTT. As I said previously, I did it over like two different time points. One of them is over 24-hour and the other one is over 48-hour. So I see that I tested it for doses between 0 to 10 microgram per ml. 0 microgram per ml is my vehicle control, and then I had 10% DMSO as my positive control. So it was noticed that for like the 24-hour treatment, there were a total of, sorry, four doses, 4, 6, 10, and the positive control, which showed cytotoxicity. But after 48-hour treatment, only 10 microgram per ml and positive control showed cytotoxicity. There was a difference in the number of doses, as we can see. So as we can see here, the 4 microgram per ml dose and 6 microgram per ml dose, it didn't induce cytotoxicity after 48-hour treatment. From what I'm expecting is, these doses are like maybe too low, and the curcumin degrades over time. So over 48-hour treatment, curcumin is definitely gonna degrade, as it's highly unstable. It's very unstable. So that is what I'm expecting. Yeah, and then there was like 8 microgram per ml. It's like an kind of an outlier. It did not induce cytotoxicity at any of the time points. It could be like due to either like errors while performing the experiment, or it might be related to a mechanism which needs further investigation. But overall, it can be said that curcumin is cytotoxic. Now, talking about my gene expression results, um, qPCR was done for TP53 and BAX genes relative to GAPDH. So um, basically interpreting the findings, it was noticed that there was no statistical difference between the doses and the vehicle control. So, like the doses that I basically induced in the cells, they did not cause like upregulation of either of the genes. From what I'm suspecting is like curcumin does induce cytotoxicity and apoptotic effects, but that might not be linked to these two genes. Maybe they are linked to different gene mechanisms which need to be further investigated. Um, yeah. Overall, the experiment was successful. Curcumin was extracted. Curcumin showed cytotoxicity in MTT assay. However, there was no upregulation of either of the genes, as we noticed. So, for my future directions for somebody who's doing like a similar kind of research would be basically choosing a different cell line for TP53. Um, it feels to me that TP53 is apoptotic. Like, no doubt, both of the genes are apoptotic, but maybe try using a different cell line for analyzing the gene expression of TP53. Previously, I read a paper for BACS where they basically used higher doses of curcumin. Like, definitely, they were more than 20, and it induced cytotoxicity. So maybe try like higher doses of curcumin for checking the gene expression of the BAX gene in the lung cancer cell line. Overall, the experiment was successful. I would like to express my gratitude to Dr. Jessica, Dr. Mike, and all the lab technicians that helped me to carry forward my research idea and fulfill my research. Thank you.